Welcome to the B191 video lecture on bacterial transformation and genetic engineering. Again, thanks to Jennifer Crossman for making these exceptional slides. What is the difference between artificial selection and biotechnology? Let's say that you have an organism, some plant or animal, that you want to adapt to your needs. You want to change its physical characteristics by changing its genetic characteristics. There are two fundamental ways that you can do this. One, you can selectively breed the organism, each generation selecting those individuals with the traits that you desire, and then breed only those individuals. This type of artificial selection is called selective breeding. Selective breeding is limited to the genes that are available in your organism. Good examples are dogs or horses that we breed to have certain traits, or household crops like tomatoes. Two, in the laboratory you can manufacture organisms by inserting genes that possess the characteristic you desire. This is broadly termed biotechnology. Biotechnology is not limited in the way that selective breeding is, because you can take genes from one species and put them into the organism that you are interested in. We're using genetic engineering, and examples are crops, pharmaceuticals, and again, animals. Biotechnology itself can be broken into two subcategories or steps. Recombinant DNA technology refers to the practice of recombining DNA, either by adding DNA segments from separate species or mixing the DNA sequence within a species. Genetic engineering is the process in which recombinant DNA that you have built or selected is inserted into an organism, thus creating a transgenic organism, or an organism with DNA from more than one species. There are 5 stages in genetic engineering. Steps 1 to 3 involve finding a gene with properties you are interested in, isolating that DNA so that you can work with it, and preparing the DNA to be inserted into an organism. Steps 4 and 5 involve placing the DNA of interest into a new organism. You will be doing steps 4 and 5 in lab, and to understand how all of these steps work together, we'll look at them in detail right now. The first step is to find the gene of interest. You have identified some trait that you need. Let's say glows in the dark. We actually get this from jellyfish. It's a real trait. Now you have to go out and find that gene. You can find the gene if you know its sequence, or you can find a gene if you know how long its sequence is. You know how long a sequence is if you can find a restriction site on either end. A restriction site being a particular sequence found on the DNA that you can identify. Sequence of nucleotides, the CATG is what I'm referring to. To isolate the gene of interest, we have to cut it out of the DNA. This step uses a restriction endonuclease, an enzyme that cleaves DNA at a restriction site. Restriction sites are usually palindromes, a series of letters that read the same backward and forward. In English, examples of palindromes include the words radar or kayak, but in genetics we are referring to the nucleotide sequences C, A, T, and G. Again, you can identify the sequence in multiple ways. Using a known sequence or a known length. But for our purposes, know that you have to cut the palindrome with a restriction endonuclease. Next, we have to get our DNA ready to insert into our organism. We use a vector to carry the recombinant DNA. The vector acts as a messenger. The vector we are going to use is a plasmid. A plasmid is a small circular extrachromosomal DNA that can replicate on its own. independent of the main chromosome. We cut the plasmid at the same palindrome where we cut the original DNA, again using a restriction endonuclease. Because we cut a palindrome, it has the same letters at each end, and we can ligate or glue the recombinant DNA onto the plasmid. In lab, you will be inserting plasmids into the bacteria E. coli. Why E. coli? Well, it's easy to work with. E. coli grows fast. It's small, doesn't take a lot of space. It has a simple genome that we can read easily, and it has circular DNA. It is an ideal organism for genetic transformation. However, we still have to take several steps to get E. coli to be competent. In other words, ready to accept DNA. One way to do this is to bathe the bacteria in calcium chloride. You see, the bacterial membrane and the plasmid are both negatively charged, and these charges repel each other. By bathing the bacteria in calcium chloride, we reverse the charge of the bacterial membrane, and the bacteria and the plasmid are now attracted to each other. Second, we can heat shock the bacteria. This opens pores and allows the plasmid to enter the bacteria more easily. Finally, we mix the bacteria and the plasmids together, allow the bacteria to replicate, and we see what happens. The last step is to check if our gene or genes have been successfully transferred to the organism. This is called screening. In lab, there are two traits we are engineering, and we will screen for both. We are trying to get the bacteria E. coli to be resistant to antibiotics. We grow E. coli on plates that have the antibiotic ampicillin and see if the bacteria survive or not. The second trait we are engineering is we will be inserting a gene from a jellyfish that will make E. coli glow in the dark. If E. coli has taken up the plasmid, it will glow under a black light. We screen the E. coli for these two traits, and we decide if we have successfully engineered the DNA of the bacteria or not.